Hello students, I am Dr. Sanskruti Agrawal. I am PhD in Biological Sciences, working as Assistant Professor at Department of Biotechnology, KTHM College. My topic of interest or my expertise is in protein biochemistry and protein structure and function. Today, I would like to discuss with you all about cell transport or cell membrane transport. So, in the image presented on my slide, you can see the image of cell membrane. Basically, you can see here that on the cell membrane there are blue color of particles, that is, there are proteins present on the cell membrane which help in transport of molecules or ions from the cell, that is from outside of the cell to inside or from inside to outside. In today's session, I would like to discuss with you all how this cell transport takes place. Before that, if you can remember, in the earlier lectures we had discussed about structure of cell membrane, that is how the cell membrane is formed or how the phospholipid bilayer is working to maintain the cell structure. Then we had discussed about components of cell membrane, that is the three types of components, that are sugar, proteins and lipids. How are they classified or how are they modified and how they maintain the cell structure by maintaining the membrane fluidity. We have also seen the different types of functions of cell membrane, and there we had discussed that major type of cell function is transport. So today let us discuss cell transport or cell membrane transport in detail. So today's session agenda would be to discuss what are transport proteins, that is there are some amount of proteins as we have already seen in the first slide, that is some proteins were present on the cell membrane which helped the cell to transfer molecules. So we'll see today what are these transport proteins or how they help in transfer of molecules from inside to outside of the cell or from outside to inside of the cell. And we'll also discuss the three types of transport, that is passive transport, active transport and bulk transport. So let us begin in discussing first what is cell transport. So the cell transport is a mechanism by which molecules or ions are transferred from outside of the cell to inside from inside to outside via the help of cell membrane, which is made up of phospholipid bilayer. So this transport is usually of two types, passive transport or active transport. So today's session we are also going to discuss in detail what is passive and active transport. But this transport takes place with the help of some types of proteins, and usually in cell membrane there are peripheral proteins present outside on the cell membrane, whereas integral membrane proteins which are embedded inside the cell membrane. So these integral membrane proteins are of two types, permeases and transport proteins which help in cell transport. So the first type of transport protein is carrier proteins. So as the word suggests, carrier means they carry the molecules from one place to another place in the cell. And they are also known as permeases because they permit or allow the molecules to transfer from phospholipid bilayer. So here, as you can see in the image, this carrier proteins or the transport proteins are present on the cell membrane, that is on the phospholipid bilayer. They bind to the protein molecules or any molecules which are present outside the cell, leading to conformational changes in the protein or the molecule, thus so that the molecule which is binding to this protein can be transferred from outside to inside of the cell. The second type of proteins work in our body to transport molecules are channel proteins. So, as the word suggests, channel means they form a channel like structure. So, as you can see in the image, they form a channel which directly transfers the molecule from one place to another. Here, the molecules or the solute which is needed to be transferred do not bind to the proteins or do not bind to these channels. Usually these channels are hydrophilic in nature. So if you can remember, we had earlier discussed that proteins are of two nature, or molecules are in or of two types of nature, hydrophilic or hydrophobic. Hydrophilic means they are water loving, whereas hydrophobic means they do not love water or they are worried to stay in the water friendly environment. So here these channels are hydrophilic, that is water loving channels. So the proteins or the molecules which are needed to be transported inside the body can be easily transported through channel proteins. So we have discussed there are two types of transport proteins, carrier proteins and channel proteins. Now we'll move on to discuss the different types of transport. So if you can remember, the first type of transport which we have seen is active transport, then passive transport. So let us first discuss what is passive transport in detail. So passive transport, as the word suggests, is just like a simple diffusion or a diffusion where the molecules are directly transferred into the cell. So here the molecules or ion move down their electrochemical gradient. That is, the molecules move from higher concentration to lower concentration. And here, as the molecules are moving according to their concentration, that is from higher to lower, here no energy is required. So this type of transport is known as passive transport. Passive transport is usually of different types based on which molecules are to be transported. There are four types of passive transport: simple diffusion, facilitated diffusion, selective facilitated diffusion, and osmosis. So we are going to discuss in detail each transport. So let us first discuss what is simple diffusion. So this simple diffusion, as the word suggests, is simple. It do not require help of anything. This type of transport takes place unaided. Unaided means it do not require help of any molecules. So here the molecule of gases, oxygen and carbon dioxide, as you can know, they can directly pass into our body, or they can directly pass to the cell with the help of or without the help of any molecules. Even the small molecules can also be transferred into our body without the help of any proteins. So this type of transport is known as simple diffusion. So as you can see in the image, there are some water molecules or small molecules that are transferred from higher concentration to lower concentration through phospholipid bilayer without the help of any molecule. So this type of transport is known as simple diffusion, which transfers the molecules from higher concentration to lower concentration, thus maintaining the equilibrium in the cell. The second type of transport is facilitated diffusion. So here, as the word suggests, facilitated means it requires the help of some types of proteins or molecules to transfer the molecules or ions from outside of cell to inside. So we have already seen there are two types of protein working in our body to transfer the molecules. They are channel proteins and carrier proteins. So we have already seen channel proteins do not bind to molecules or ion. They directly transfer the molecules from one part to other part of cell. Whereas carrier proteins bind to the molecules, there is conformational change in the molecules or the conformational change in the proteins, thus transferring the molecules from one part of cell to other. So as here some specific molecules are required for transport, this type of diffusion is known as facilitated diffusion. Usually these channel proteins which are present in our body are made up of glycoproteins. Glycoproteins means proteins which are modified with sugars. So here in this case, in this type of transport, rate of transport of molecule is higher as compared to any other transport. The third type of transport is selective facilitated diffusion. So as the word suggests, selective means here selective molecules or ions can only pass through the membrane. So these channels are specific for certain molecules or ion, and as you can see in the image, there are different channels or different channels or channel proteins or carrier proteins to transfer specific ions like potassium, sodium, or calcium. Similarly, the last type of transfer or last type of passive diffusion is osmosis. So, you would have already heard about osmosis. So, the major difference here is that osmosis is takes place through semi-permeable membrane. That is, here water molecules are transferred from higher water concentration to lower water concentration through semi-permeable membrane. So, as you can see in the image, here they have taken two sugar solution. One solution is highly concentrated and the other one is low concentration sugar solutions. For an example, here consider if you mix uh, 2 spoons full of sugar in 1 glass of water and you mix 5 spoons of sugar in 1 glass of water. So when you allow to come this sugar solutions in contact through a semi-permeable membrane, you will see that water molecules will pass through lower concentration will pass through lower concentration of sugar, that is will pass through high concentration of water, that is the sugar solution which is having 2 spoons of sugar will have more water concentration. So the sugar water will pass from higher water concentration to low water concentration. It is similar as you can see in the image. Water molecules are passing from low sugar concentration, that is high water potential, to low water potential. So here we have already discussed the four types of passive transport. The first one is simple diffusion, second selective facilitated diffusion, and facilitated diffusion, and lastly osmosis. Let us now begin to discuss about what is active transport. So it is somewhat reversible to the passive transport. This type of transport requires transport proteins. And here, transport proteins means pumps. Different types of pumps work to transfer ions or molecules for from inside of cell to outside or from outside to inside. Here this type of transport requires metabolic energy, that is in form of ATP. The energy in form of ATP is required to transport molecules in active transport. So there are different types of pumps working to transport molecules like calcium pump or proton pump. So let us discuss what is proton pump. So proton pumps are different pumps working to maintain the concentration of different ions into our body. So one best example is sodium potassium pump. The sodium potassium pumps works to maintain works to maintain the equilibrium concentration of sodium inside our cell. So usually you know that concentration of sodium is much higher in blood as compared to that in our cell. Usually in our cell, it is l- m- near about 10 millimolar, whereas outside our cell, it is in the blood, it is 150 millimolar. Whereas composition of potassium is exactly reversible. That is, potassium is found more inside our cell and less outside, that is in blood. So this lower concentration of sodium is maintained in our body with help of sodium potassium pump. So let us discuss the mechanism of the sodium potassium pump. So if you can remember, I told you that sodium potassium pump or an active transport require energy. So here ATPase enzyme works to convert ATP to ADP, and when the energy is liberated, this there's a change in the concentration, that is there is a release of the sodium ions from inside of cell to outside and taking up of potassium ions from outside to inside. So in this type of pump, as you can see in the image, this sodium potassium exchange pump, which is an electrogenic transmembrane ATPase, is present on the cell membrane, that is phospholipid bilayer, and it pumps 3 sodium ions out of cell with the simultaneous transfer of 2 potassium ions inside the cell. So this type of transport, or when the transport of ions takes place inside the sodium potassium pump, these ions are transferred against their concentration gradient. That is, they are transferred from lower concentration to higher concentration. So in our body, sodium potassium pump maintains the lower concentration of sodium inside the cell. Next, we'll move on to discuss the third type of transport, that is bulk transport. So, as the word suggests, bulk means when the molecules are in larger size, that is macromolecules. So, you already know there are some macromolecules present in our body which have different functions. So, these macromolecules can be transferred inside the body through help of exocytosis and endocytosis. Bulk transport takes place in our body through exocytosis and endocytosis. So, as the word suggests, exocytosis means macromolecules are secreted out from the cell by help of exocytosis, whereas they are ingested inside the cell or they are taken inside the cell from outside by endocytosis. So, let us begin to discuss in detail exocytosis and endocytosis. So, exocytosis is also called cell vomiting. As the word suggests, exo means to give out. So, here in our body, there are different proteins and molecules synthesized in our body, specifically inside the cell, and they are needed to be transported outside to reach their specific location or to reach the area where they function. So, in our body, specifically, proteins are synthesized in ribosomes. So, the proteins from the ribosomes are transferred to endoplasmic reticulum and through endoplasmic reticulum to Golgi bodies, which sort and transport the molecules to their target site, that is through the cell membrane. They are transported outside the cell and they are transported wherever they are required to function. So, here the secretory vesicles are present in our body, which transfer this protein molecule synthesized in our cell from inside to outside. So, here this exocytosis do not work only to transfer the proteins, they also work to transport some amount of waste material or the extra material which is not required by the cell. So, as you can see in the image here, the extra material forms a vesicle like structure. This vesicle fuses with the plasma membrane, and the molecules are given outside from the cell membrane. So, this type of mechanism of exocytosis also help to vomit any molecule which is not required by the cell or any waste which is not required by the cell from inside to outside. The next type of bulk transport is endocytosis. So, usually, endocytosis takes place in reverse manner to exocytosis. That is, here the molecules are taken from outside to inside of the cell. So, here, if you can see in the image, that is plasma membrane, the membrane which is present on the cell surface, folds inside so that the molecules come inside this fold and forming a vesicle, these molecules are taken inside the cell. So, here the plasma membrane folds or invaginate so that the molecule is taken inside the cell. This endocytosis, depending on the which type of molecule is transferred inside the cell, is of three types: phagocytosis, pinocytosis, and receptor mediated endocytosis. So, in phagocytosis, large molecules are taken inside the cell. So, as you can see in the image, that the plasma membrane is invaginated, solid particle, the green color particle comes inside this plasma membrane, the invaginated plasma membrane, which forms a vesicle known as phagosome. And this phagosome goes and fuses with the lysosome. And you know that lysosome has hydrolytic enzymes which cleave this solid materials, thus trying to eliminate this solid or harmful materials which are not required by body by killing through the hydrolytic enzymes. Mostly this function of phagocytosis is performed by macrophages in our body. The second type of endocytosis is pinocytosis. So the pinocytosis is taking up of liquid particles. It is also known as cell drinking. So here what is done, that is extracellular liquid material is taken inside the cell, again in the same manner, that is the plasma membrane invaginates, liquid molecules are taken inside the cell and they form a vesicles which can go and fuse again with the lysosome, or they can go to the place wherever they are required. The third type of endocytosis is receptor mediated endocytosis. As you can see, or you can see here, the word receptor is important because specific molecules can enter the body or enter the cell only when they have some receptors helping them to come inside the cell. So here the specific receptors are seen on the membrane to which the specific proteins or specific molecules can bind. So this receptor mediate binding is similar to the substrate and ligand binding, so that when there is any molecules present outside binds to this receptor, they form a zome or a vesicle which is taken inside the cell. So here the vesicle formation can be coated with some types of proteins or cannot be coated with the proteins. So usually here two types of vesicle are formed: coated and non-coated vesicles are formed, which help in forming the vesicles. So usually here endocytosis is of three types: phagocytosis, pinocytosis, and receptor mediated endocytosis. So here we end with what is bulk transport. So bulk transport is of two types: exocytosis and endocytosis. So before ending our session today, I'd like to give you some assignment. I would like you all to differentiate between passive and active transport. That is, write a difference between passive and active transport, explain what is sodium potassium pump, and also try to explain with help of diagrams what is endocytosis or types of endocytosis. Let us summarize today's session. So here today we were discussing about cell transport, how cell transport takes place, what are the different proteins help in cell transport, and we had seen basic three types of cell transport, that is active transport, passive transport, and bulk transport. We had seen that active transport requires energy, that is, here the molecules are transferred in opposite direction to their concentration or against their concentration gradient. So usually in active transport, different types of pumps work to transfer the molecules or ions. Example are sodium potassium pump and calcium pump. The second type of transport is passive transport, that is opposite to active transport. It does not require energy. Molecules are transferred from higher concentration to lower concentration, and it is of four types depending on which molecules are transferred: simple diffusion, facilitated diffusion, selective facilitated diffusion, and osmosis. And the last type of transport which takes place in our body to transfer macromolecules is two types: exocytosis and endocytosis. So exocytosis is transferring the molecules from inside of cell to outside, which is known as cell vomiting, whereas endocytosis is transferring the molecules from outside to inside of cell. And again we had seen that endocytosis is of three types depending on which molecules are transported inside the cell, that is phagocytosis, where solid particles are transported; pinocytosis, which is known as cell drinking, where small molecules or the liquid particles taken inside the cell; and last is receptor mediated endocytosis, where some specific molecules bind to the receptors and they are transferred inside the cell. So this is all about cell transport. So we have discussed what is cell transport, how do the cell transport takes place with help of different proteins, and what are the basic three types of transport. In next session, we'll discuss what is cell signaling. So here we'll discuss what are the signal molecules working to transport the molecules from one position to other, or activating the cell to bring about some certain mechanisms. We'll see what are the different types of signaling, which are the signaling molecules, and we'll also study receptor mediated cell signaling. These are the few references which you can refer to studying in detail about cell structure, cell membrane structure, as well as cell membrane transport, types of transport, including what are the proteins useful for transport. So with this, I end my session on cell membrane transport. I hope it was beneficial to you all. Thank you.